to come and sit in the front, Professor Huang and Professor Hu. Okay. So now we have 20 minutes for discussion. Our first session by Professor Wilson talked about the value of metagenomics in diagnosing neurologic infections, particularly TB meningitis, recurrent meningitis of unknown causes, and viral meningitis, and ended with a cautionary tale from Mayo Clinic, highlighting scenarios where false positive may occur, and the importance of sending samples that are from an initial CSF tap and avoiding testing asymptomatic cases. The second session from Professor Chu talked about the role of metagenomics in diagnosing challenging infectious diseases to improve patient outcome and lower healthcare costs. And they gave examples of vaccine-related rubella encephalitis and others. And also the application of metagenomics for diagnosis of infections using body fluids and plasma. And lastly, he talked about host response classifications to detect and predict certain diagnoses. So now we are open to the questions from the floor and from the online audience. Is there any questions from the floor?
I have a question for Professor Chu. You mentioned two interesting cases. One is the hemophilus in the lung, in a case of AML. Another case is the most interesting is fulminant hepatitis in children in the era of COVID-19. But in your result, you show that the virus or the bug is there. But can you prove that they are the causative agents for the whole disease? For example, for AAV2, do they need any other help? Or is the virus alone sufficient to cause the whole picture? And how do you differentiate the situation and to convince the people that they are the most important etiology?
Yeah, that's a fantastic question, and I think it really highlights. I think your question really highlights the potential limitations of metagenomic sequencing, because ultimately what we're doing with metagenomic sequencing is we're detecting the DNA, we're detecting nucleic acid, and unfortunately it's not a functional assay in the sense that it doesn't tell us necessarily, it doesn't tell us that what you detected actually is causing the infection. And so this is particularly relevant with the pediatric hepatitis cases we looked at, because in fact, all of those cases, it was not just adeno-associated virus 2, it was actually one or two additional viruses. And indeed, in cell cultures, and we do know that adeno-associated virus 2 traditionally typically requires a helper virus to successfully infect cells. And so, for instance, for adenovirus, for AAV2 to infect liver cells, it needs to have another virus that basically facilitates the infection. So the other virus is commonly thought to be potentially an adenovirus or herpes virus. And indeed, we actually did detect an adenovirus and a herpes virus in all of those cases. But what was quite interesting was that one thing is that the AAV2, for instance, we detected that in liver tissue. So, and we detected it actually in the viral load, which I didn't actually tell you, but the viral loads in liver tissue were about 10,000 to 100,000 times higher than that of the other virus. Now, that still doesn't prove, of course, that AAV2 is actually causing the disease. But I do think it's actually quite interesting that there is a clear association between AAV2. And so I think that potentially there may be an analogy with another hepatitis virus called hepatitis D. And I know that many of you infectious disease clinicians in Taiwan are well aware of that. In the hepatitis D, by itself is not thought to cause disease, but in combination, if it co-infects the liver with hepatitis B, then you can end up with liver failure, a very fulminant liver presentation. So ultimately, the analogy may hold true for AAV2 as well, but certainly further work is needed, probably animal models, cell culture models, and epidemiology. So further investigation is really needed to directly prove causality. And so the test really doesn't answer that question, and you really need to do further work to be able to establish that.
Okay, thank you. I believe Dr. Hu has a question.
I'm Dr. Hu. I have a question for Dr. Wilson. You showed wonderful data about transcriptomics for increasing of sensitivity of CNS infection. I'm wondering what is the next step you will apply this technique in the clinical practice? Thank you.
So I think the next step is, this collaboration that Charles Chu is leading, looking at, we had a very specific question in the TB study. This was a population of HIV infected patients in a TB endemic area. And so the big question there is it TB, or if not, what else could it be? And so that was the focus there. I think in the United States, oftentimes the most challenging patients for us in the hospital are encephalitis patients, are those who have a CSF profile with maybe 20 to 50 cells, many of which are lymphocytes, and they may or may not have a fever, but they certainly have confusion, seizures, and in those cases we go back and forth between whether this could be an acute viral encephalitis or an autoimmune encephalitis. And so I think in the US, that's I think the time where neurologists and other specialists, I feel most helpless, and we know that patients with these autoimmune conditions can have very rapid and dramatic recoveries with steroids, and that their ability to recover is very time dependent. And so I think those are the cases where having a negative mNGS result and a host response that indicates that it's less likely to be infectious would really help build the confidence of treating physicians to start empiric immune suppression, or if it turns out that it really does look like there's an occult infection there, maybe withholding those for a bit longer. So I think that's what I'm most excited about next. I think, in the Uganda work, also building up the number of cases so that we can say more than just TB versus not, and again, get more detailed, like Charles discussed, looking at subcategories of diagnosis.
Oh, thank you. Is there any more questions from the floor? If not, there's a follow-up question that's online. It's question number 2 and question number 6. It's concerning, Dr. Wilson was talking about TB meningitis, and the question is concerning whether higher reads can increase the sensitivity to detect tuberculous meningitis, and if there's a certain number that you would suggest. That's question number 2.
So I think the answer is yes. Sequencing more deeply will always, will increase your sensitivity for any pathogen. Certainly, that's the case for TB. I think it's hard to put an exact number on kind of, because of course the sequencing depth also depends on the specifics of the kit that's being used. Different kits have different performance characteristics, how you make your sequencing libraries, how much CSF you're doing extractions on, do you spin the spinal fluid down beforehand, et cetera. So there's many other technical factors that go into, for a specific protocol determining what the optimal sequencing depth is. So I don't have a specific number, but certainly we were still picking up very low level TB detection, at 50 or 100 million reads. I don't think that's necessarily economical or feasible to do that, to scale that up. But yeah, TB is hard to find. I'll say that.
Thank you. I think we have a question from the floor, please. Please identify yourself. Thank you.
Hi, Charles and Michael. This is Yao Ting from National Zhongzheng University, and I have two questions for both of you. The first one is a more general question, because now we are aware there are many large panels, such as Illumina respiratory pathogen panel, or many large panel developed by the mainland China company that could enrich hundreds or thousands of pathogens. What's your comments or your experience on using those panels prior to metagenomic sequencing? And the second question is about a little bit technical issue, because when we are trying to do RNA sequencing on the host response, we always suffer a problem of very low biomass or very low RNA quantity collected from the spinal fluid. What's your opinion? Do you still do RNA depletion even for those low quantity samples, or you don't do RNA depletion prior to metagenomic sequencing? Thank you.
Yeah,
Go ahead, Charles.
You want to take it, Michael? I can.
No, go for it.
Okay. I can perhaps start by answering, and then I'm certain that Dr. Milson will be able to add on this. But I think with regards to your first question about the use of kind of probes to enrich for targets, that's definitely a viable approach. It's a great. There are some advantages to using probe-based enrichment. One is that because it's typically a smaller subset of targets, it becomes, in theory, it's probably easier to validate the approach, for instance, for regulatory agencies. So if you want to run it, say, as a clinical test versus running it as a research use only test, there may be some fewer hurdles to using enrichment, because, for instance, there actually have been several panels that are now FDA approved, at least in cancer, for instance. So, and the other benefit is that you can vastly improve sensitivity. You can improve sensitivity because you are enriching for a subset of pathogens. And as you mentioned before, now many different companies are producing these very large probe enrichment panels. There are, however, several disadvantages. So one disadvantage is that because you're narrowing your focus, you probably have less sensitivity for detection of, say, novel or emerging pathogens, potentially for things that are not specifically targeted. Viruses, for instance, have quite a bit of diversity. So if you're using probe enrichment panels, they may not be able to detect, for instance, highly divergent viruses, or even viruses which are well known, like HIV, which can be highly divergent. The HIV, for instance, has like a 50% divergence in the envelope gene. So you may lose some sensitivity for metagenomic detection. The other issues are primarily logistical, in that typically the enrichment by itself takes about at least 8 to 24 hours, in addition to producing libraries. So it becomes quite problematic to run this routinely in the clinical lab. And then the other point is that it can be very expensive. So for instance, like Twist Biosciences, which is a US company, produces these panels, but to run 100 samples for the probes alone, it's $5,000 US dollars. So it's something which may not be cost effective, at least this current moment. And I think that probably the last disadvantage would be that you can't really do these other types of analyses. Like if you want to look at host response, for instance, if you want to look for, say, markers of cancer, because you are biasing your sample by enriching for pathogens, you kind of lose the flexibility of being able to do these additional analyses. And then for your second question, don't actually remember. Do you remember, Michael, what the question was?
Oh, it's just how do you get good transcriptomic data from such biomass samples?
Yeah, great. Yeah, so that's a great question as well. So in practice, we still do ribosomal RNA depletion, and the reason we do ribosomal RNA depletion is that's not very useful transcriptome analysis. So we want to get rid of the human ribosomal RNA. When we tried to actually also get rid of bacterial ribosomal RNA for, say, bacterial contamination, that's when we noticed that there was a loss of sensitivity. So our current protocol, which is only based on human ribosomal RNA depletion, but we are able to recover actually fairly good yields from very low input samples. But Dr. Wilson has a lot of experience. So Michael, do you want to comment?
I'm very sympathetic. I think that to this problem. I think that where we've had some success is, I think the New England Biolabs sequencing kits that we switched to a few years ago have provided, I would think, overall smoother and more comprehensive coverage in the RNA-seq data sets that we generate relative to kits that we had used in the past. I think that we also switched a couple of years ago to a QIAGEN-based, it's called a Fast Select ribosomal RNA depletion kit, and that had in our hands, that's really kind of zeroed out the ribosomal RNA in the CSF samples, and so that I think has also helped us with getting better coverage across the transcriptome. What else? Spinning down the samples, increases the yield to some degree. And I think it's some combination of all of these things, but it's certainly and obviously getting high quality samples. I think training, getting the clinical community aware that these tests exist and that they may be useful in their cases, and understanding that that these samples are going to be much more informative if they're frozen quickly. And I think that's maintaining the cold chain for these samples obviously is incredibly important too. So it's not any one thing, but it's a combination of things that has over the years kind of helped the yield that we've been able to pull out of these samples.
Thank you. A follow-up question, question number 8. There's a question concerning if the samples were in enclosed spaces like Dr. Wilson talked about, CSF, it's fine. But for complicated microbiome in diversity samples such as tissue and sputum, how do you differentiate between colonization and infection? I think Dr. Wilson, if you could talk about this.
I'll let Charles talk about that because he deals with the respiratory. I think spinal fluid's difficult because there's not much in there, but I agree that the colonization issue is quite complicated in respiratory samples. I'll let Charles take that.
Yes, both respiratory and stool samples are diarrheal samples are very difficult because they have an endogenous microbiome, and so in the healthy state, and in fact, it becomes very difficult to detect. We essentially detect kind of with colonizers. We detect pathogens, but we also detect, for instance, like reagent contaminants. So the way we deal with it is that so the way we deal with reagent contaminants is that we implement very, very strict or very stringent internal and external controls. So these include both internally spike controls as well as external spike controls, which are run in parallel with every single set of clinical samples. But to deal with the colonization issue, the test itself won't tell you whether anything's a pathogen or not, and so that's why I think we typically provide the results as is, and then we leave it up to the clinician to provide clinical interpretation. So it's really I think that it basically highlights the fact that you still need to take these results into clinical context, the results that you get from the test. I mean, we would like the test to be able to provide all the answers, but I view it as simply another diagnostic tool. Now, it's a very important diagnostic tool, but it's still another diagnostic tool. And really, whenever you evaluate a patient, and consider managing or treating a patient, you really want to leverage all of the data that you have available, which includes the data that you get from the patient's history, the physical, the laboratory testing, the radiological testing, as well as the results from this particular test. I do think in the future, though, there are two advances that may help us, at least with respect to respiratory samples. One is that is the use of host response, as we've been discussing earlier. There's another colleague of ours, Dr. Chaslin Jellier, who from UCSF, who has done a lot of work with looking at correlating host responses from respiratory fluid with the pathogen detection. And that way it can provide context and can help you determine, is what you detected really a colonizer or a pathogen. The second thing that can be done from both stool samples as well as from respiratory samples and other samples as well is you could do a microbiome profiling analysis. So this is commonly done for research. But one thing that we've noted and that others have noted is that in the setting of a true infection, it severely perturbs the microbiome profile, so you don't see as much diversity as you might see in, say, someone who's healthy. So leveraging microbiome data, leveraging host response data, and as well as the metagenomic detection data, I think would be the way to be able to tell the difference between colonization and pathogen. But ultimately, in the end, you want to take all of these findings, like all laboratory tests in clinical contexts.
Now, I'll just add one thing and say that although I strongly believe that there is no microbiome in the central nervous system, and Charles can attest to this, that when you look at a metagenomic data set generated from a spinal fluid sample, it looks as if there is a central nervous system microbiome. And there are some papers out there in which I think people have misinterpreted those data sets and alleged that there is a CNS microbiome and that people can even have a dysbiosis in the setting of multiple sclerosis or other diseases. And so that highlights a different problem, which connects to a previous question about the low biomass in spinal fluid, and I believe is also the case in bronchoalveolar lavage fluid, which is that when there's very, very little there to sequence, speaking in terms of low picogram quantities of RNA and DNA, then we've found that there's an overrepresentation of the environmental contaminants in the sequencing that are present in the kits from the sequencing reagents or even in the sterile water. And so I think it's spinal fluid is simpler in the sense that it doesn't have there's not a microbiome in the body, but there is a microbiome in CSF metagenomic data sets, and to remove that or account for it, you really need to, as Charles said, have rigorous controls, including no template or water controls, to understand kind of the specific environmental microbiome that's in your lab and in your kits, because you can very easily go astray.
Okay, a related question to host response, question number 7. Maybe Charles can help with that. It's talking about whether host response can predict a prognosis of the infection. Can it be a predictor of patient recovery? Charles, maybe you could answer that one.
Yeah, so in fact, it's a very interesting question. And we, in fact, are doing some work in that. We're doing work on, can the host response be able to, not only potentially diagnose an infection, but be able to provide prognostic information. So, for instance, can the host response kind of early in the course of infection be able to predict kind of the direction a patient is going, whether or not a patient will improve or whether the patient is gradually deteriorating. And so we are looking at that. The big problem, I think the big challenge is that many of these host responses appear to be quite distinct, even between related organisms. So the challenge has been in, can you apply that broadly across metagenomic range of organisms, and I think the answer is we don't really have the data right now, but we are looking at, for instance, host response and infection prognosis for certain conditions. So one is like COVID. I'm very interested in SARS coronavirus 2 and COVID infection in terms of can we determine, can the host response at the early infection, can it predict kind of the overall prognosis or the clinical course of the patient? And we're also looking at it with bacterial sepsis in a way of host response in terms of whether that can predict the course of, which patients will deteriorate, will have to be transferred to the intensive care unit, and who would improve with appropriate antibiotics. So this is a very interesting question, and I think that there will be a lot more information over time as we get more metagenomic data. So currently, I feel that it can be a predictor, but probably only with specific infections we have enough data.
Yeah, and I'll just echo that and say that's actually the next thing that we're focusing on now in the TB meningitis study. So half of patients survive and half of patients don't, and many of these Ugandan patients are being enrolled in therapeutic trials where they are getting serial lumbar punctures. And so the kind of next set of data we're generating now is looking at exactly that, as to both at time point 0, whether the host profile predicts the half of patients who survive the hospitalization and half that don't, but also just change in CSF profile over time.
Thank you. There's a question maybe for Dr. Wilson. Question number 1. Is it possible to use host transcriptome to be a biomarker to help to diagnose clinical infection?
Well, I mean, if I understand the question correctly, I think we would both agree, absolutely, that we've tried to highlight cases where that's been in disease states where that's been helpful. And again, I think I see the utility both as helping contextualize a negative metagenomic result to feel more confident that you just didn't miss an infection, but that it truly is a non-infectious disease, but also maybe to increase the sensitivity for picking up a hard to detect infection. And then lastly, as Charles showed a case, viral or autoimmune cases where likely contaminants were detected by mNGS, to give confidence that when a suspected contaminant is detected, if the host response doesn't fit with a bacterial infection, to build more confidence that there really was a contaminant and not a strange infection someone might have.
Thank you.
Yeah, I'd also like to add that using the other application of host transcriptomic analysis is that it could potentially guide the development of more focused or condensed panels. So the idea would be that what you detected, the specific differentially expressed genes that you identify, could be leveraged in that you could then envision developing a portable or rapid kind of multiplex RT-PCR assay, for instance, where you could run hundreds of clinical samples at a time in a way that's faster than you could do by sequencing, at least currently. So that would be, and we actually have some collaborations where we're doing exactly that. We're taking a subset of markers, say 30 to 50, that were identified by host transcriptome analysis, and we're putting that onto a platform where we can run it in a high throughput fashion.
Thank you. Due to the time constraint, I will have to close this session. And I'd like to thank again the two speakers, Dr. Wilson and Dr. Chu, for their wonderful talks, and Dr. Hu. Okay, thank you. Now we'll hand over the moderating to the next moderator, Professor Lu.